In this video, we will continue with the computational modeling of host pathogen interactions. So, we already have a Boolean model, building the model is the hard part, and now we will go about simulating this model and deriving interesting insights, even from by deleting nodes, perturbations as usual and so on, to understand the important components of this host pathogen interaction machinery. How do you now simulate? So, as we studied in the previous class, we can either go in for a synchronous update or an asynchronous update. When does one go in for a synchronous update versus asynchronous? So, asynchronous gives you a lot more stochasticity in process duration, right. You can have processes that are relatively more fast. Synchronous assumes that everything takes one time step to happen, right, be it antigen presentation or the macrophage binding to some cell, whatever. All that takes exactly one time step, whereas the asynchronous allows for some more stochasticity. You can even have like longer term memory. You can even depend on something that was like 5 steps previously, 3 steps previously and all that, although we did not really do that here. So, we also tweaked the Boolean model to account for delay in the onset of adaptive immunity. You have an adaptive immune system, but that never kicks off on day 0, right. It kicks off only after some punishment has been received from the bacterium, right. And what is the initial bacterial load? Then when does persistence set in? How efficient are we clearing the bacterium? This can depend on drugs and so on. And is there a delay in phagocytosis, apoptosis and so on and so forth. So, we made essentially a semi-quantitative model for this, and if the growth rate of bacterium is taken as lambda for exponential growth, you can think of a simple equation like this. So, Bt is B naught into e to the lambda t, and we can update the population in this fashion. So, we can say Bt plus 1 is 1 plus alpha into Bt, and solving this you can see that alpha is nothing but e to the lambda minus 1. Okay, this is very simple. So, we essentially used alpha as an input parameter to the model, and so alpha is nothing but the incremental change in bacterial population at each time step. If conditions are good for bacterial clearance, right. So, what does bacteria growing and clearing mean as far as the Boolean model is concerned? True and false, right. So, if bacteria star evaluates as true, I will try to use this equation to grow the bacterium. If bacteria star evaluates as false, I will use this equation to basically clear out the bacterium, right. So, this is like a clearance efficiency and so on, which depends potentially on any drugs that are being taken and things like that. So, we used an asynchronous update, and we also had ranks for the Boolean functions to enforce update of certain nodes before the others. We saw that how that works in Boolean net in the previous lab. So, these are all the parameters that we used. So, we looked at an initial bacterial load, and this is the possible range that we determined through a study of literature and so on. So, B naught, then what is the maximum bacteria for infection to like really be irreversible in some sense, then what is the threshold for clearance, and how long should it persist, how long should it stay in the body within a zone before it enters persistence, and what is the, so this is the number of bacteria that should be there, and this is the time. So, 14 is 14 days or 2 weeks. This is onset of adaptive immunity, which is again 2 weeks. Then some default values for bacterial growth and clearance and delay in phagocytosis, apoptosis and so on and so forth, and we simulated for 15 weeks. It is a sort of macro model. It is not a very cellular model. In some sense, it's a cellular model. It takes into account the cellular components and so on, but then overall looks at the concentration of bacteria in the whole human sort of thing, right. So, we performed the simulations using Boolean net, and we had 3 possible disease outcomes. So, it could be either active disease, persistence or bacterial clearance, and we performed 1200 runs of the simulation, and the dominant outcome we found was persistence, which is actually what happens in reality, right. TB, most people are just carriers of TB without actually even manifesting, what we found that for example, we did find that we can sort of mimic vaccination by putting delta AI is 0. So, that is the adaptive immunity delay. So, what does it mean when delta AI is 0? It means you have already been exposed, right, which is what happens during vaccination, and we find that in almost all cases they do not go towards active disease, which is reasonable observation, but of course sometimes you do find that vaccinated individuals in reality can contract disease. So, we find that low initial bacterial load, we have more persistence, and high initial infection you have more active disease, and again if you have low clearance or low growth, persistence is predominant, and high growth active disease and high clearance efficiency you have bacterial clearance and so on. And although these are like sort of fictitious parameters in the model, you can think of alpha and eta to be influenced by any drugs that are being taken and so on. So, if you are taking rifampicin, that would have a particular influence on alpha and eta. If you are taking isoniazid, it may have a different influence on alpha and eta, and so on. So, and this is like a state map for the simulations. So, this lists several of the 75 states that we have, right. So, so blue is on and white is off and so on. And if you see, I have not shown bacteria here because it makes more sense to look at bacteria as the load, right, because we have a discrete update for bacterial population after each time step. So, this is how the bacterial population varies. So, after this point of time it basically goes into persistence. And we also, as in any systems biology experiment, one wants to do perturbations, right. What happens when there is some defect in the system? If there is a bacterial protein that is not working, perhaps because of a drug, or there is an immune defect or so on, for whatever reason. So, what are the, or even without going into the physiological significance of these experiments, it helps us understand what are the key players in these systems, right, and what are the key drivers of these interactions. So, this is a map of knockouts, and which show what fraction of simulations resulted in active disease. Right. So, if you see, if I knocked out phagolysis formation or phagocytosis, it always resulted in disease. You see a completely dark band there, and if inflammatory molecules are not present in the human body, it means your defense is too weak. So, in a lot of cases it was leading to active disease. So, this is along with, so this is a double knockout, this matrix. So, it shows you what happens when I remove inflammatory molecules and cathelicidin. So, two major inflammatory molecules are out, disease always sets in, right, and so on. So, this again, as I was mentioning, reflect the critical components of the system. So, anytime you knocked out phagolysosome formation and phagocytosis together, it always led to active disease. And we also found that most knockouts lead to a higher incidence of persistence. That is not visible from this figure, but it is visible from this figure. The figure on the right shows, you can see it is a very dark plot, right, compared to the others, right. So, the darker the square is, more fraction of simulations ended in that outcome, which in this case is persistence. So, except you see a full white band for phagolysosome, because all of this is in the previous slide where it all went to active disease. So, if you see this, you find that whenever CAT G is not there, whenever CAT G is knocked out, it almost goes into persistence, right. And similarly, whenever apoptosis is knocked out, persistence is more common, and so on and so forth. So, you can study what are also, when IL-10 is knocked out along with there are some pro-inflammatory and anti-inflammatory cytokines, right. So, there is a delicate balance between these two in the body. So, you see that sometimes it can result in clearance, a lot of times when this is knocked out, and in many cases when apoptosis is knocked out, there is no clearance at all, or when interferon gamma is not there is practically no clearance at all, right. So, these are very interesting insights that one can obtain by studying knockouts or perturbations on these kinds of models. So, to summarize, this just gives you a bird's eye view of the host pathogen interplay. It is a far more complex network that underlies this. This is just but a small very high resolution, or a high level view of what happens. But the model is amenable to various analysis such as node deletions and so on. It gives you good insights into critical immune processes, and it's a very strong framework for integrating quantitative data. Right? If you know what is going to be the effect of a particular drug on a particular component, you can easily include that and so on. But obviously the model is limiting in the sense you currently have only on and off states for practically everything. We did put something for the bacterial population and so on, but otherwise it's just either on or off. But this you can imagine is a sort of a strong way to look at a nice way to study host pathogen interactions. You can study metabolism as well, but the good thing is that you can study metabolism and integrate it into this model, right? Because this model presents you a lot of opportunities to integrate, as I was mentioning even in a previous class. The Boolean framework or the discrete framework gives you an opportunity to integrate from other kinds of modeling techniques. You can like solve an ODE to decide the outcome of a particular state, right? Or you can solve an FBA model to predict the outcome of some other state and so on. So there are 75 nodes including bacteria, 18 virulence factors. These are actually from the bacteria, right? So these are bacterial cell and bacterial molecules inside the bacterial cell. 56 host components including 11 cellular processes, 19 cells and cell states, and 26 molecules themselves. In this video we looked at simulations of the Boolean network model that we built for studying MTB human interactions. It was not a pure Boolean model, but we also had some nice hacks to include how the bacterial load changes and the efficacy of a drug and so on. And in the next video we will switch gears and look at we will start looking at some advanced topics. We will first look at robustness in biological systems. I will give you a brief overview of robustness in biological systems.